So now it is time for us to move forward to the first talk of this meeting. Let's welcome Aviv Regev, the spiritual leader of the Human Cell Atlas. She pioneers the use of single-cell genomics and other technologies to dissect molecular networks that regulate genes, define cells and tissues, and influence health and disease. Welcome.
Thank you very much, Guojin. It's a pleasure to be here virtually and in spirit. And I couldn't agree more about the fact that when we come together with science, knowledge, data, and collaboration, we really can conquer very difficult challenges. And so I'm going to give a brief introduction to the Human Cell Atlas in general, talk a little bit about where things stand in the Atlas, and also share a couple of scientific stories from Atlas research in the context of human disease. I have to start with my disclosures. I have equity in Celsius Therapeutics and Immunitas. Until July 31st, I was a consultant for Thermo Fisher, Neogene, and Syros. And since August 1st, 2020, I'm an employee of Genentech and Roche. And so, as you know, the mission of the Human Cell Atlas is to create comprehensive reference maps of the types and properties of all human cells, which are the fundamental unit of life, as a basis for us to understand, diagnose, monitor, and treat in health and disease. And we sometimes fondly call it a periodic table of our cells. But aside from the beautiful analogy, I think one of the things that many of us hope is that, similar to the periodic table of the elements, that wasn't just a nice way to chart all the elements, it actually highlighted the organizing principles and a theory for thinking about chemistry, that studies of our cells would allow us to understand the principles underlying cellular properties in the human body. Human Cell Atlas has really kicked off in October 2016, and spent its first year mostly as a planning phase and also setting up a lot of the infrastructure and getting together as a community to articulate our vision and kick things into high gear. And since October 2017, we have been in a very active collection mode, and so we're really getting close to closing our third year for that. During this time, through many meetings, networks that have formed, and other activities, the Atlas is being collected, and at the same time, I think we all share in our scientific knowledge and expertise and really advance both technology and biological and clinical insights forward. And so a brief snapshot into the status of the HCA community. As you know, HCA is a completely open global initiative. Anyone who is willing to adhere to the values of the HCA is welcome to join. And we have just crossed the 2,000 member mark a couple of days ago. In fact, yesterday night, it was 2,003 members from 75 countries and more than 1,200 institutes, and 99 of these members are in China. That's as a little tribute to Guoji himself, across 74 institutions. And in fact, the people involved in HCA are a much larger number than that. Often within a single lab, one person from that lab would register as a member, but actually all members of the lab participate. And I urge those who are not members to join the HCA. HCA scientists are organized in different ways, and one of the ones that has really emerged now as a major principle for us are biological networks that span the different organs and systems of focus of the HCA, including organs such as, the brain or the gut or the heart, but also systems such as the immune system and particular phases of like human development. And as we will hear later, during this virtual meeting, also tackling questions in genetic diversity. And scientists actually from HCA China have played really critical role at nucleating and leading many of these networks. Our overall goal is to build a comprehensive atlas over a 10-year period, and we are well on our way for our first draft of about 100 million cells. And just like this graphical rendition, even though we have partial information as we build the atlas, even the partial information is incredibly useful for us. So just as a quick snapshot of where we stand across about 12 of our systems, in many of these systems, here are the liver, the kidney, the skin, the lung, the gut, and human development, we have substantially passed the 1 million cell mark, and in almost all cases, the 500,000 cell mark. And we have also analyzed samples from dozens and sometimes hundreds of individuals. That actually indicates to us that we're getting from a census perspective, just knowing the cells that are there, to a point that's going to get close to saturation for many of the organs, and that can assure us that we're already recognizing also very rare cells. At the same time, this community has really sprung into action around COVID-19, and in many of these organs that are actually highly relevant to COVID-19 patients, such as the lungs, the kidney, and the liver, we also have cells now that have been profiled from COVID-19 patients, and I will come back to that. To show you six of the additional organs, in particular in the brain, there is a fantastic effort that is led especially because of the Brain Initiative and has crossed the 10 million cell mark, but also in systems that in the beginning of HCA were not at the forefront, such as reproductive systems that are really giving us a very substantial glimpse and level of information. And finally, in the immune system, which is one of the more accessible systems for us for single cell analysis, we're well on our way to the 7 million cell mark. And again, in the relevant tissue, also information from COVID-19 patients, especially in PBMCs. And I believe that Sarah, who is going to talk later in the symposium, is going to cover that aspect. I also wanted to give a glimpse into part of our mission, which is to use the reference atlas as a context by which to understand not just health, but also disease. And the two are really inextricably coupled to each other. If we know what health looks like, we understand disease better. But also by looking at disease, we actually understand what homeostasis and health looks like as well. And in the context of a disease of a disease study, there's a series of questions that scientists are interested in, like where might disease risk genes from human genetics act, which cells might be disrupted, how are their cell programs changed, what happens to their functions, how are the cell communications disrupted, what might be the effect of drug. And it turns out that a healthy reference atlas can be very useful for many of these queries. So I'm just going to give you a glimpse into one particular query, which is maybe the simplest one that you would ask of an atlas, which is, where is my gene? And in that context, the Human Cell Atlas actually has developed a really close partnership with human disease atlases, both globally and through specific initiatives. And so if there is particular initiatives that support healthy reference studies, they work now really hand in hand with human disease studies. And I want to highlight in particular the Lifetime Initiative from Europe that has really taken a strong disease focus complementary to the HCA. And so in the two examples that I'm going to show you, I'm going to talk about common disease and how we can do queries for genes to really help us understand better genes from genome-wide association studies. And then I'm going to turn and talk a little bit about COVID-19. And so for my first example, I'm taking the context of the gut cell atlas and how we might try to use it in order to understand a complex genetic disease like ulcerative colitis, which has really been a poster child of human genetics because it has allowed us to identify many new causes and in many cases highlight new pathways that we didn't ever suspect would be involved in ulcerative colitis. But for many of them, we do not know the cell of action, and we do not also know the process in which the genes participate. And so amongst the cells that I've shown you for the gut, which have now well past the 1 million cell mark, about 265,000 of those cells were collected in a study that looked both at healthy tissue and at samples from ulcerative colitis. And in this context, we can develop an atlas, and this atlas combines both the healthy and the disease cells together, embeds them together, and says 51 different subsets of cells, in this case, only in the colon mucosa, which is one layer of the colon from both healthy individuals and 17 individuals with ulcerative colitis. Doing this and having a healthy reference allows us to ask the most simple question about disease. Has something happened to its cells? And we find that there is a dramatic remodeling of the gut cell composition. And we can only do that because we can compare the disease tissue to the healthy reference atlas. And in this case, we might find that there's M cells that are present only in ulcerative colitis and typically are not actually prominent in the colon at all. There's these fibroblasts that assume this inflammatory state, which we again do not see in healthy colon. And then there are these T cells that express both CD8 and IL-17 that are greatly expanded in ulcerative colitis. Now, if we turn to the genes from genome-wide association studies, given that we have this beautiful catalog of 51 subsets of cells, we can ask very easily, where might they be expressed? And so we're focused here on those genes that are actually, where the genetic signal was already narrowed down to a single gene level. And we look at them in the columns, and we consider their expression across all 51 cells in this colon mucosa atlas. We can see that we can identify cell type specific expression both in healthy and ulcerative colitis, irrespective of the condition. There are genes that are expressed specifically only in healthy tissues, and there are genes that are expressed specifically only in ulcerative colitis. Now we can go to a particular cell type, such as these M-like cells that are expanded in the disease, and we can look at their enrichment for genes that come from GWAS. And we see that there is a preponderance of genes from genome-wide association studies that are actually expressed in M cells, and moreover, those genes are induced in those cells under inflamed conditions. So this is a very simple but effective use of an atlas, but there's something that I didn't tell you. And so I showed you the genes that have cell type specific expression. There are about 50% of the genes that were already mapped to a single gene level in the locus. But what about the other 50%? Now I'm showing them as well. They're to the right of the plot. And you can see that those genes are either not very specifically expressed or honestly not that much expressed at all. And so there could be many reasons for this, including interesting biological ones. But the most prominent one is that we didn't actually profile the whole colon in this initial atlas. It was only the mucosa layer, which is only one layer. There are cell types like adipocytes, for example, that are only present in deeper layers like the muscularis layer. In addition, there are some cell types that are still missing from this atlas, and in particular, very rare cell types like enteric neurons, which are not just rare and so harder to capture at large numbers, but they actually do not survive dissociation of the tissue into single cell analysis. And so we have recently developed two lab protocols. One is called RAISIN-seq that allows us to do better single nucleus RNA-seq, which allows us to capture neurons, and the other called Miracle-seq for label-free rare cell enrichment in order to get a more comprehensive view of the tissue. And when we do this and apply it in this case to frozen resections, healthy portions of the colon, we obtain another 150,000 profiles, amongst which are, for example, 1,500 enteric neurons, but also other rare cells like interstitial cells of Cajal and adipocytes. And when we do this and we go back to the same plot that we looked at before, it now turns out that many of those genes that were not expressed at all or expressed lowly or not specifically in the mucosa have very specific expression patterns inside these additional layers. They might be expressed in adipocytes, they might be expressed in subsets of enteric neurons. And moreover, now we can start and look at other types of diseases that might not affect the colon mucosa, but for example, where we expect to see an effect in the enteric nervous system because there is a gas dysmotility phenotype, such as a rare disease like Hirschsprung's disease, or common diseases that are typically central nervous system diseases like autism spectrum disorder and Parkinson's disease, but that have a gut dysmotility comorbidity. And so this really highlights to us the importance of systematic characterization of atlases. It's not enough to have a portion of the human body because genes are everywhere and you shouldn't assume that they're necessarily where you expect them to be. So here I really want to give a shout out to Guoji. This is his human cell landscape rendered on something that looks a little bit like the globe. So we thought that was cute. But it's really one of the first examples that we have of a cross-tissue atlas, where you can start asking these questions without assuming that you're necessarily in the right tissue, because maybe the gene is actually important in an additional cell type in another tissue. And most recently, we have deepened this kind of approach, in this case, in the context of an autopsy study, looking at 8 different tissues and examining, in the end, 37 different cell types across these tissue subsets, seeing both substantial commonalities between tissues as well as a lot of tissue-specific expression, even in shared cells such as immune cells, stromal cells, and so on. Most recently, we have started taking this approach one step further, trying to combine the single cell signal together with the human genetic signal in an analysis of heritability to try to say which cell carries sufficient heritability signal for particular diseases. We developed this approach together with Alkas Price, who's a human geneticist, and now we can look at the heritability for inflammatory bowel disease, a signal coming from human genetics, and cross it against the atlas. First, looking at the healthy cells and their cell type specific expression. We see, for example, myeloid cells, which is well appreciated for this disease, but for example, also these M cells that before were not known to contribute to disease risk or barely were known to even be there in the colon. You can also do it now with gene programs that are specific to the disease conditions, such as in ulcerative colitis, and now you find the fibroblast-associated disease program that carries heritability for the disease. And finally, you can do this systematically for each of the cell types, asking both on the cell type and on the disease condition, and you see that enterocytes are important in IBD overall, and especially in ulcerative colitis, whereas ILCs are important in IBD and Crohn's, but not so much in ulcerative colitis, trans-amplifying cells, the opposite, and T cells across the board. And so the second quick vignette that I would like to give you has to do with how we use the atlas to understand a disease when it's emerging, when we don't have, hundreds and thousands of papers in the literature as we have for inflammatory bowel disease, but we're all living through it in our own backyard and in our own neighborhoods and communities. And of course, this is COVID-19. It's a beautiful story also of our community coming together. And so there's many different questions that are and still are open in the context of COVID-19, including transmission. Sarah will speak to that in her talk later on, the epidemiology of the disease, why we see it hitting some subsets of the population more than others, the pathogenesis, the disease mechanisms, and also the viability of model organisms, which we need in order, for example, to develop therapeutics. And I like the example of human genetic studies in the context of the Human Cell Atlas. The first question that one would want to ask is, where are the cells that the virus might infect? And here, of course, by February, it was already quite well established that ACE2, which is the receptor for SARS-CoV-2, along with it needs an accessory protease, TMPRSS2 and cathepsin L, and possibly others could be those receptors, and we can use those genes as a very simple query across the atlas. I won't belabor this. This is published work, but or posted work on the bioRxiv. But the critical piece for me to emphasize is that this was an international effort across the entire Human Cell Atlas community. It really started with the lung biological network because our original focus was, of course, the lung and the airways. But as our understanding of the disease evolved, as well as just our curiosity of the possibilities increased, then actually people from the heart network and from the liver network and from the kidney network and so on have all joined in. From the initial study, we have just from the Healthy Atlas, we have learned a lot. We have learned about cells involved in transmission, about the potential for transmission through the placenta and the fetal lung, so vertical transmission, the different tissues in which pathogenesis might occur, which actually track extremely well, unfortunately, with the pathology that we see in patients, in the involvement of pre-wired immune-like programs that are in the cells that are likely infected, different epidemiological aspects, including the association with age, sex, and smoking status, as well as the suitability of model organisms, both the mouse and non-human primates. But as time moved and as we all are living through this experience, then our same community really rallied around, in the spirit of science, knowledge, data, and collaboration, our community has really rallied around studying with single-cell technologies and spatial technologies patient samples. There are studies of the blood, of other specimens that you can obtain from living patients, such as aspirates, nasal swabs, and lavages. Sarah will speak to those. And in addition, there are multiple efforts focused on autopsies in order to really access tissues in the depth of the body where the virus is really wreaking havoc. And I will focus on an autopsy study like that for the next 5 minutes or so. And so there are several such efforts. I'm highlighting two on this slide, one coming from the Boston area, and I'll focus on that one. The other coming from Imperial College London. There are also efforts in New York, multiples, in Arizona, and in multiple locations in Europe. In a typical study, such as this one, this would be considered a relatively large one. These are, of course, very difficult studies to conduct. This one has studied 17 different patients across a broad range of ages, different types of organ failure that lead to death, from lung to multi-organ failure, in one case neural complications, and a collection at least of lung, heart, liver, and kidney in almost all patients, and then from a subset of the donors, different subsets of the tissues. And for those who live in the United States, the health disparities and the high impact on African Americans and Latinx individuals is really emphasized in this table. What do we do in a study like this? Well, we apply our Atlas toolbox. All that we have learned to do in other settings, we can apply to COVID-19. We can obtain fresh tissue and try to process it to single cells. We can obtain frozen tissue and process it to single nuclei. And we can obtain fixed tissues in FFPEs and perform spatial analysis. And we've actually done all of those things. So first of all, to emphasize the great challenge and difficulties for scientists to do this. So these are 3 heroes who worked in the BL3. 2 of them have never worked in a BL3 before this pandemic, but they were experts in single cell and single nucleus RNA-seq. And the third was a BL3 expert, but has never done single cell studies. And these types of studies start in the autopsy suite, in the hospital, and through a very rapid style of communication and little kits that you invent on the fly, they end up in a BL3 facility for processing. You also need to set up the right processing timelines. These are pipelines. These are far from perfect studies. So for example, we have to, these autopsies sometimes are conducted more than 24 hours after death. And so there's plenty of ambient RNA in the tissue. We partnered with the cell vendor developers in order to remove this ambient RNA. You have to annotate almost a whole human cell atlas cross-tissue study in a very short amount of time. So we developed a transfer learning approach for that. And you also want to study where the virus is, so you have to have a system to analyze viral RNA at the same time. And without belaboring all the technical steps, this is an example of the lung portion of the study, 70 lung autopsy samples, almost 37,000 cells. You can see the nice overlap between the patients. You can see the annotations, and you can see both some repeatability as well as quite a bit of variation in the composition of the different samples. Now, of course, you can go back to the context of the healthy reference, and just like the UC example that I showed you, you can ask, how does this compare to healthy tissue? And also because Atlas studies, such as in Naftali Kaminsky's lab, are also looking at other lung diseases, such as in this case, idiopathic pulmonary fibrosis, you can compare also to other types of diseases. And we see that across the 3 major compartments, stroma, immune, and epithelial, the COVID-19 lung looks different from both IPF and healthy lung, but also the same is true also when you dig into finer annotations and the compositions of the cells. You can also start asking questions in the context of human genetics. So in parallel to this effort, there's an international effort to conduct GWAS of COVID-19 severity. Several of the loci have already been mapped, for example, chromosome 3 locus and chromosome 21 locus. We can now look at the genes in these loci and try to help the human geneticists in the same way that has been done in UC in order to know in which cells are those genes expressed in a relevant tissue, such as the COVID lung, as well as in the healthy lung. Now we can start digging in, and we can ask about expression changes in each subset of cells. So for example, 82 cells, which are a major target of the virus, you can ask, what is the difference in expression profiles of 82 cells in COVID-19 lung versus healthy lung? And we find reduced expression of surfactants and reduced anti-apoptotic activity, both of which are consistent with the phenotypes we see in patients, and an induction of inflammatory, innate immune damage response, cell migration, and ECM pathways in the cells. Along doing such studies, you can actually reach some meaningful scientific insights. So for example, one of the things that we found is this enrichment of a gene program known as PATS for pre-alveolar type 1 transitional cell state, which is a gene program that gets activated when 81 cells, which are responsible for oxygen exchange, when they're damaged and there isn't enough stem cell capacity to replenish them, 82 cells can transdifferentiate to 81 cells. They do it through a transitional state, and sometimes they can get stuck in this state. This can happen in fibrosis, and it seems to happen in COVID lung as well. Another aspect of this is a change in the in the fibroblast compartment, and we're still looking into that. Finally, you can ask, where is the virus? And so for this, we use viral reads. We take our annotated atlas, we look at the entry factors, and we actually find that the viral reads, by the point of time that we're looking at, which is when people are unfortunately deceased, the viral reads are not actually in the cells that have the preponderance of the entry factors. And this can be for various reasons. For this, you need to know in which cells they are. So we can relate viral RNA reads to ask about whether viral RNA reads are related to cell infection. We ask about alignment to the minus strand versus the plus strand. We have some indication that it might be related to it, but we cannot definitively say that this is productive infection. There is dramatic variation in the detected viral RNA in the cells across patients, and we find that macrophages and vascular endothelial cells have the highest number of cells with viral RNA. And as we drill into these two subsets of cells, in macrophages and endothelial cells, and compare cells with and without viral RNA, we see that they express different gene programs, which are actually consistent with a response, at least to viral RNA, not necessarily to viral infection. Such as, chemokine-mediated signaling, type 1 interference signaling pathways, and so on. So finally, for the COVID-19 story, I want to turn very quickly to looking at it also with a spatial perspective, because of course our ATLAS studies always have both a cellular and nuclear arm and a spatial arm. And so first, just to give you a glimpse of what a COVID lung might look like, in pink is actually SARS-CoV-2 probe. This is with RNA scope, and you can see the very high level of infection in this particular lung, but you can also see that it can be very specific to particular parts and doesn't actually go into other adjacent parts. So in order to get a nuanced view of the spatial microenvironment, and since we had to do this work actually with fixed tissue in FFPEs, we turned to the nanostring GeoMX DSP, which allows us to first identify a region of interest and then specifically capture the RNA from an area of interest within that region of interest. This is an example from patient SO3, and on the right at the bottom you can see the mask that we apply in segmentation and capture only the RNA from the mask from the region of interest. When we do this, we can take then each of the profiles that we obtained from each segment, an epithelial segment on the left, a non-epithelial mass region on the right. We can compare we compare here 3 healthy lungs to 7 COVID-19 patient samples. And we can take the profile from that region together with the cellular profiles we have from the single nucleus analysis and infer the cell type composition for each region of interest. And by this, we find that there is a reduction in mast cells and an increase in macrophages and myofibroblasts in COVID-19 versus control samples. Now we can go within, compare patients and controls, and we can compare the COVID-19 high patients to the controls and identify the pathways that are activated in the alveoli in both epithelial and non-epithelial cells, and find different types of both interferon alpha and interferon gamma and other immune responses. We can also ask within the same patient to compare COVID high versus low regions of interest, which are both epithelial, and identify a unique response to the infected cells that we don't see in the non-COVID infected cells, which are epithelial and in the same patient. And those involve, again, interferon alpha and interferon gamma response and other antiviral responses. Coming up, liver, kidney, heart, and additional atlases from the same patients, which would also allow us to compare within the same patient for different things. And as I said, Sarah will touch on other aspects of this major effort in the community. We have set up multiple resources, including in partnership with the Lifetime Initiative, to allow the community to register their interest in this kind of research and to find partners, experimental, computational, clinical, to understand the data that we're all collecting. In the last couple of minutes, I want to turn back to why we're able as a community for HCA to pivot so quickly. And this is actually built on all of the work that we've done and are doing in the Human Cell Atlas initiative over the past several years. We were ready to go because we were collaborating already, because we had infrastructure, platforms, methodology, and an extremely strong sense of openness and community, starting with our values. And within them, I want to really emphasize community equity, our transparency and open data sharing, and our commitment at the same time as we study human samples for privacy and ethics. These things that really permeate through our working groups. So in our analysis working group, led by Dana Peer and John Marioni, in addition to organizing data analysis teams, meetings, and jamborees, some of the methods, such as the ambient cleaning method, is an offshoot of a jamboree that happened quite a while back. But also recommending experimental designs and analysis frameworks. The data coordination platform at data.humancellatlas.org, where we leverage a data store that has built-in support for managed access, which is coming up soon, but not just there yet, and that will have a data access committee to facilitate access to controlled access data, which would really improve our ability to do human studies. And the cell annotation platform, I already highlighted the annotation as both a major challenge and one of our greatest opportunities to come together quickly, which is led by Peter Karchenko, working together with Fabian Theis and John Marioni. The lab protocols that I highlighted, such as all of those nuclei protocols, are really embraced by the efforts of the Standards and Technology Working Group, the STWG, led by Holger Hein, Orit Rosenblatt-Rosen, and Zhouwei Zhuang, spanning both single-cell and spatial methodologies, and having gone through multiple cross-community comparisons of protocols, yielding more and more papers along the way that really allow all of us to move better and faster. Our equity working group, our most recent working group, but an activity that actually started in 2016, led by Musam Langa, Partha Majumdar, and Alex Shalek, is really committed to promoting and supporting progress towards equity in the HCA. In the COVID-19 context, this played a critical role in the ability to really compare to other infectious disease, because this is something that the equity working group has been committed to as part of its global efforts. And it works not just to ensure that our atlas is diverse in its science, that our samples are diverse, and I believe Jay will speak to that, but also that it is diverse in its scientists and in where they are in the world. This has to be a truly global effort. In June, in the context of both local events in the United States and more globally, we came up with a strong statement on our commitment not just to Black Lives Matter, but also to global ethnic equity. And we identified specific actions, including our current commitments to expand outreach programs in underrepresented countries, especially LMICs, to implement regular diversity audits for our membership, our leadership, our study participants, and our collected data, and address the gaps, and to work with funders to encourage the earmarking of funds to increase recruitment of scientists and study participants for underrepresented regions and communities. And I'm thrilled to say that the CZI has actually very recently announced a supplement that it would provide its grantees in order to meet this specific action. In addition, one day when we travel again, we will have travel stipends. In fact, the virtual world has improved our equity standing, and it's something to really appreciate. And we're in the process of running a survey for equity, as well as have done at least one equity roundtable to brainstorm additional solutions. And finally, our ethics working group, led by Bartha Knoppers, Barbara Wald, and Roderick Luijo, and really facilitated by Emily Kirby at McGill, working with Bartha, allows us to make sure that what we do really serves humans. It doesn't just serve our science, it serves the patients, it serves the individuals, it serves the people who so generously donate and agree to participate as research subjects, without whom there would be no human cell atlas. And so they focus on an ethics toolkit, developing launching our ethics help desk, and ensuring that people from across the world can really support. And so I hope I gave you a glimpse today of where the Human Cell Atlas stands, of our commitment to bring together the community and our science. No time like the present reminds us how important it is to have data-driven approaches to biology. Mysteries still abound, and how important it is to be global, open, and collaborative. We're excited to see the steps ahead as the Atlas grows and becomes more and more complete. And with this, I will finish and be happy to take any questions that Goji sends my way.
Okay, fantastic talk, Aviv. Can everyone use the Slido to type in your questions? So we have a question from Zeming now, and he's asking, hi, Aviv, in your COVID-19 studies, did you detect viral sequences in non-epithelial cells? What are they? How real are those? And do those cells with viral sequences mean direct infection?
This is an excellent question. I tried to touch on it, but I really was a little short on time. First of all, they're real. They are real, they are significant, they are not ambient RNA. That is one of the things that we have devoted a substantial amount of effort for, because we wanted to be certain. And we also know that when we bulk them up, we actually get the result of bulk viral sequencing. So we have another additional control that I didn't even touch on. Their preponderance in macrophages needs to be taken carefully, not with respect to their realness. They are absolutely real, but they can be the result of phagocytosis. And it is very difficult to rule that possibility out, because the one tool that we have in our arsenal using sequence to try and distinguish things, you can look at basically some version, right, of transcripts that you would expect to be present only in a replicating virus. And here is the difference between the minus strand and the plus strand for SARS-CoV-2, and others have done it with splicing. When you do that, you do see some signal that is indicating of a virus, I would say, in the process of being replicating. It is a much lower signal, which is to be expected, but it is substantially lower. And moreover, it still cannot rule out in a macrophage the possibility that it phagocytosed a cell that was not a macrophage in which there was a virus that was replicating. So we are extremely cautious about concluding that this is a signal of replication. There are in vitro studies from other groups that have shown that as you progress in the very early stages, there is a signal like this in the minus versus plus strand, but we can't extrapolate it. And especially because these are such late stage, I mean, these are autopsy samples, these individuals have passed away, we're surely not seeing the first wave of viral infection. In the endothelial cells, which is the other major cell type in vascular cells, vascular endothelial cells, which are the next major cell types in which we see reads, it's a similar story. And we did not feel the confidence to say that this is productive infection. Having said that, it is absolutely where the reads are. It's not an artifact.
Okay, so Aviv, I have a question actually. I'm still, continuous, always asking myself. So in the human genome projects, people do sequencing, and every base is a base. But for our, human cell atlas project, and there can be lots of variations in digestion and data quality. So how, what we can do to improve those, the integration of the ultimate atlas?
So that's a great question. First of all, I want to say something about the human genome and base errors. Every base is a base in reality. Sequencing makes a lot of errors. We all know that. People knew it then. Coming up with quality metrics was not just it wasn't an obsession. It was absolutely justified. It took years and years and years. It's not that when the Human Genome Project started, everyone got perfect base calls, and errors existed and errors subsisted. And depending on the sequence that you were sequencing, there were regions that took many, many years to finish after we had a draft human genome in 2000, and there's regions that are not finished to this day. There are still gaps. So first of all, there's the completeness and the error question, even for the human genome. In the human genome, you have a second layer, which again, I think is really timely for this meeting, which is the aspect of human genetic variation. The Human Genome Project didn't answer the question of human genetic variation. It provided the scaffold on which human genetic studies could act. So similarly, I would say for us, I think we could actually go a little faster and a little better than we originally anticipated, because we got the technologies better and they got improved with time. The question you ask, though, is a different one, because it's a question that asks about the variation between cells, which is driven from technical aspects versus the variation that is driven from non-technical aspects. One of the things that is very comforting, or increasingly comforting, at looking at these studies that now span dozens of labs, hundreds of samples, millions of cells, is that in fact, when you do all these lung studies, they kind of agree with each other. They're not radically different from each other, even though there are differences. There are differences between doing nuclei and cells. There's differences, of course, between the regions that you actually study in the lung. But one thing that the COVID-19 effort made us do more rapidly than we might have done otherwise is that we got this huge view of more than 20-something studies by now of the lung. And when you look at all of these 21 studies of the lung, and they weren't even processed together, not just experimentally, they were processed computationally by their own researchers. There's a lot of limitations on what was on what could be done in such a short time. When you look at them, at the end of the day, once you control for the fact that not all of these studies sampled the same region, studies of the same region with the same strategy gave pretty much the same results. So I would say, just like human genomes, there's variation, but by and large, it's pretty resilient. Our computational techniques got a lot better. Our experimental techniques got better too. So by now we're in a pretty solid space. And one of the benefits actually of the way that the atlas is being constructed by multiple groups doing the same organ in parallel but coordinating through a network is that we have a real sense of this variance. Because if everything would have been done in one place, maybe everything would have looked really clean and tight, but some of it would actually be systematic error, rather than it would be the error unique to that. And that's actually a lesson learned from sequencing studies, that you actually don't want to do them all in one place because each place has its own set of errors.
That's a great answer. Thanks. So because of time, we have to move to our next speaker.